Good morning, everyone. This is Peter Moole, and I'm going to share with you our fourth webinar on real-world drug effects, looking from the perspective of the More Europa project. And a warm welcome. I'll go and share my screen. This. And if everything is right, you should be seeing my screen. So we are going to talk today about understanding real-world drug effects by performing federated analysis. And we're going to use the case example of the Mora Europa project. That is that we are looking in 4 multiple sclerosis patient registries. Just for housekeeping today, we will mute all participants automatically, and we do not have enabled the chat box, but ask you to put your questions in the Q&A box. We will not always be in the position to respond to questions or comments during the presentation, but we'll have plenty of time at the end of the webinar. It is being recorded, and we will also share that recording later with you all after the webinar. So this is the outline of today. First, I will give you a bit of a context of the project and what we want to do with federated analysis, or where we believe that is important. And then Lars Forsberg will talk about the case example in MS registries and really take you in a long motion, but a really active motion through 3 aspects of this federated analysis. One more around the disease mechanism of MS and why we want to intervene there. A part on really understanding how federated computers actually work, and then tell you more about the project's approach to using data from different registries. And Mariana from the Radboud University will talk about Bayesian federated inferences. How can we really make sense of these data in an efficient way at multiple sites? Going a little bit more into the technical discussion around this. And then finally, we will have some time to interact with you based on questions that you may have. And this is being moderated by Mariana Gildmark. So this is the 4th webinar. As I started out saying, we had already in November 23 on generally the use of registries in regulatory decision-making. In July 2024, we talked about AI and machine learning approaches, really to identify the wealth and the breadth of registry data around on the globe, and how to identify which ones contain relevant data, bringing you to the machine learning tool that we have developed with the More Europa project, and that will soon be launched. In February, we talked more about the integration of real-world data into clinical trial data, and to help understand these outcomes of trials better in the setting of clinical practice. We are 4 people today. I am Peter Moole from the University Medical Center Groningen, a professor of drug regulatory science, but also a regulator sitting at the CHMP committee of the EMA, where we really take decisions on approval of medicines. Mariana is a colleague sitting on the advisory board of the Mora Europa project, and she holds a master's in economics and policy sciences in Copenhagen, but also has a Master of Philosophy in Health Economics from York University in the UK. And she leads change strategies focused on EU, the European Union, HTA, Health Technology Assessment Organizational Readiness, because there's so much going on at this stage. And we're very pleased that Mariana wants to share our discussion later with you. She's also sitting in Basel, Switzerland, at the Global Value Market Access and Pricing Group, and she's a member of the International Society of Pharmacoeconomics Outcomes Research, and served various roles in the board of directors of that. And very nice, I recently visited Copenhagen, and I think it's a very nice role to also be a goodwill ambassador to that. Lars Forsberg is then our main speaker of today. And he is a very interesting guy, I would say. You will certainly agree with me after you've heard him presenting. He's an engineering physicist with a PhD in neuroscience, and he's working currently at the Karolinska Institute, focused on real-world data, working with the really nice and extremely well-organized multiple sclerosis data set. And Mariana Jonker will then dive deeper with you in the approach to Bayesian federated inferences, and she has studied mathematics and obtained her PhD in mathematical statistics at the Vrije Universiteit, the Free University in Amsterdam, and she's currently sitting at the Radboud University Medical Center in Nijmegen, another major city in the Netherlands. And she focuses her research on these federated inferences and survival analysis, and is really the scientist among us going to talk about these statistical physics, mathematics, and integration with medicine approaches. So that is the speakers of today, and I hope you're really looking forward to the presentations, and I am myself, but taking you back briefly to where the More-EUROPA project is about, and that is really a Horizon Europe sponsored project that is focusing around two main questions that are currently in play of, well, the promise of real-world data, can it be used to support regulators and HTA in their decision-making? So one hand, establishing the value, really understanding what is in those data. In our case, we look at registry-based real-world data in really helping understand outcomes of clinical trials, and also enabling more effective and ethical use of registry data to support patient-centered regulatory and HTA decision-making, really including views also on how can we use those data? What do patients feel? What should be done with these data? And how do we really effectively integrate that in clinical practice? And this is graphically a depiction of how the project looks like. In work package 1, we have the novel analytical tools, and you're going to hear especially about the federated analysis that we're talking about today, in which we are working on to really make sure that we can establish value. Can we trust and understand what comes out of these data? In work package 2 is really the core of the project, where we have the 3 data registries and 3 case studies that we use for all our understanding of what to do with those kind of data, which is in heart failure, multiple sclerosis, and lung cancer. So those case studies will help us understand how these data may then be started to be used. And we use also to enforce, let's say, the enabling, making it more feasible to use this kind of data. We have a screening, we have developed a screening tool to identify suitable registries. I talked about the machine learning tool webinar. And we also really have dedicated work package looking at ethical and patient perspectives. For instance, can we use can we make secondary use of these data, and what do patients feel of that? What are the ethical aspects around this? But also really explaining in simple videos, for instance, to patients what we are doing when we are talking about real-world data, and having a set of webinars there having been developed by the European Medical Multiple Sclerosis platform. And then today your part are following on the work package 5, where it's really about training and the option and use of all that we discover and identify in the project. Also from other projects, trying to integrate how to move forward and put these learnings into the practice of regulators and HTA, and also obviously other stakeholders that want to make use of this kind of data. Another graphical depiction, mostly focusing and telling you that we are looking across different spectra of the drug life cycle, so pre-licensing, licensing, and post-licensing. Focus in this project primarily on efficacy and effectiveness, cost effectiveness, and making a set of tools that can be used to integrate real-world data and randomized controlled trials. You see the registries that we have, and we also intend to work towards guidelines and train HTA and regulators on how to combine real-world data and RCT. data, randomized controlled trials data. So at the core, as said, 3 registries, 2 from Sweden, the Swedish Multiple Sclerosis Registry and the Swedish Heart Failure Registry, and in addition to that, the cancer lung from the Netherlands. All established for at least a decade, but the Swedish registry is already for more than 2, and numbers ranging from 10,000 to over 150,000 patients. The interesting bit of the Swedish registry is obviously the extensive linkage to other data sets that are possible through the unique patient identifier. And here are some other characteristics important to see in terms of age range captured, but also the differences in male and female, for instance, in those registries where you do see that diseases sometimes have a different representation or prevalence of the disease. Important, obviously, to realize. And finally, one of the aspects is that we talk often of real-world data in contrast with trials, but there's actually sort of continuous. We can do case control studies, we can do purely observational work, but we can also look at randomizing clinical practice and then follow up these patients using clinical practice outcome data. And that is, for instance, done in this REFUND and the SPIRIT-HF trials, which were randomized, but patients followed in clinical practice. We will hear more about REFUND today. And this is, for instance, to give you an idea of that fantastic Swedish data linkage possibility. This is the Swedish heart failure with all of its linked kind of data sets where we can follow the cause of death really through these individual identifiers and in principle the dispensed drug registry. So we really have these data without doing all kind of manual searches, and that makes obviously these data much stronger and reliable. And there we come to the core of the discussion these days. We have the data quality framework from the EMA, and key concepts are that obviously a registry needs to be having a sort of foundational thinking around what is quality in the sense of how to generate, collect, and expose these data over longer periods of time. But some things can only be really addressed by a specific questions. And there is also the middle layer, describe the intrinsic aspects that talk about things that are inherent to a specific question, but not really defined by the question. So they need to be there, but they will not need to perhaps new data generation. And then there are 5 dimensions identified. Are the data sufficient? Do we have enough? Are the data, can we analyze it? Are they coherent? Do we get the data in time? Often a question for regulators and HTA is really, we have to make a decision now based on a safety aspect, based on a decision to reimburse, and that should be, can we really get the data when we need it? Very important also is the relevance. Are we having the right type of data? And there I want to come to the aspect of also our federated analysis of today. And these are, for instance, important differences in the epidemiology of disease, even in the EU. We are talking about More Europa, so it's a project really aimed at the European regulators, not saying that these findings cannot be extrapolated to other settings, but primarily focus at. And what you see here is the incidence and prevalence of heart failure. And where between blue and red, you see a twofold difference in these incidence and prevalences. And you can see the countries on the map. Equally, there are important differences also in disease management. And these are, again, between blue and red, up to twofold differences in heart failure-related hospital discharges or duration of hospital stay. So these are really important differences that will also affect, if you perform a study in one of these countries, in one of the data sets available, that this may affect this outcome and the observation that you make. If you have a relative assessment of outcomes, it may not necessarily harm, but it will affect, obviously, the absolute outcomes that you observe and also perhaps the feasibility of studies. Randomized clinical trials are often performed in multiple settings and therefore the sort of ameliorate, let's say, these differences in population outcomes and disease management. So for the EU regulator and HTA, we now do have this data set called Darwin EU, where lots of data are being collected in a standardized kind of way, using a common data model, and where in the meantime, 39 data sources are combined from 30 data partners, capturing 181 million EU inhabitants from 18 countries. And what is specific in this data set is that the data stays local, and at the orange box, circle in the middle on the right-hand side, there's a coordination center that sends out questions, and the analysis are performed by these data partners. And that is the way how Darwin EU works. It doesn't mean that all data are there for all 18 countries, but at least there is information in different sets, and sometimes some or more of these data partners can contribute data to answer the questions that are being asked by the EMA and its committees. So this is how the EMA performs. And now I would like you to listen to my colleague Lars Forsberg, really talking about the content of how we in More Europa work with federated analysis. So I'm gonna stop sharing my screen and look if there's already questions. Otherwise, I don't see any questions currently. So I think we can really move on to our first speaker, Lars. Please, the floor is yours.
Oh, thank you very much, Peter, and thank you for that introduction. Just see that you can see me like this, I hope. Just checking my setup for you. So good. Yes, so I will talk about federated analysis using MS registry data in Europe. I just want to I just want to mention my colleagues, my group leader Anna Glaser, my previous group leader Professor Jan Hillert, my colleagues Elena Moresan and Bo Bekush, that are also working in the Mora Europa project. So federated analysis it's not just one specific method, but it's a number of different methods to be able to run statistical analysis and machine learning algorithms between data sources, but without merging data. This is the key thing with federated learning, because nowadays we are not able to share individual patient level data even between our MS registries. It's becoming more difficult due to patient safety. So we need to use these new methods to be able to run things like logistic regression that I will talk about today. And so the case study that we have for federating learning in MS are between 4 different BMSD registries in Europe. It's Sweden, Italy, Denmark, and Czech Republic. So BMSD registries, these are registries that are part of the big multiple sclerosis data network, consisting of these 4 registries, as well as France and the international MS-based registry. So this network together has a total number of patients being 350,000 patients. So I would like to go back in time to the year 1868, because it was this year that Jean-Martin Charcot established, through a number of series of lectures that he had, multiple sclerosis as a disease of the nervous system. And he also recognized that it may follow a progressive course. So Charcot is often considered to be maybe the first neurologist, and he did not only look into MS, but also Parkinson and epilepsy and other brain diseases. Of course, he didn't have an MR scanner back then, but he looked at the brains after the patients had died, and he did see these lesions that were very visible when he looked at the brains from these dead patients. So he saw that there were damaged myelin in the brain, and today we know that this is due to acute inflammation in white matter, and these damages are causing lesions in white matter, and some of these lesions may result in what is called relapses, which is a clinical manifestation of those lesions. Then, as I also mentioned, he also recognized that there were also a progressive phase in MS, and today we know that this is due to chronic inflammation with chronic active lesions, so-called dream lesions, and chronic active microglia. And also neurodegeneration. So overall, we now know today that MS is caused by it being a chronic autoimmune disease where the immune system attacks the myelin in the white matter, causing lesions in the brain or in the spinal cord. The prevalence of MS is around roughly 0.2%, at least something like that in Sweden. And I guess it is similar in the other European countries as well. So not all these lesions actually cause a clinical brain dysfunction. Some lesions are not even noticed by the patient, but over time, of course, as they accumulate, it can get worse. But when a lesion causes a clinical brain dysfunction, so that, for instance, the patient is not able to walk that well or having problems with vision and so on, depending on where these lesions appear in the brain, this is known as a relapse. And MS patients with relapses, they have what is called a relapsing remitting MS course. The brain may then recover from these relapses after some months and regain function, sometimes not to a 100%, but close. But some damages may also remain, and actually 70% of all lesions remain as scars in the brain. But the brain is also at the same time a very plastic organ and can compensate for those lesions as well, at least up to a point. But after some years, the disease may develop into a progressive course independently of these relapses, and this is then called a secondary progressive MS. And until today, there has not been any effective treatments available for this secondary part of the disease, but there are now coming new treatments, so-called BTK inhibitors, and there are great hope that these will be able to at least reduce the progression. Some patients get even this progressive MS from the start, and then it's called primary progressive MS. And after some years, these MS may lead to a wide range of symptoms: fatigue, visual disturbances, walking difficulties, and cognitive issues, etc. So if we then move forward in time to 1955, it was this year that Douglas McAlpine, he formally described these three major MS subtypes to be then what we now call relapsing remitting MS, secondary progressive MS, and primary progressive MS. And it was also the same year that John Kurtzky developed a scale called the disability status scale, and he further developed it in 1983 to become the expanded disability status scale. And that scale is what we still use today in clinical trials and in research. This is a scale where a 0 means normal neurological function, and then it is going in steps of 0.5 up to, well, in all 10 means death, so the highest a living patient can reach is 9.5. And this scale has been very useful to understand the disease course. So it is thanks to EDSS that we can also understand and separate between these different subtypes. It would take quite many years, but in 1993 or 95 in Europe, the first disease-modifying treatment became available for MS patients, and it was soon followed by more treatments. These first treatments, they were not great, but they were better than not having any treatment at all. So they wouldn't reduce all the relapses, remove them all, but at least reduce the level of number of relapses. In 1998, Mabtera, also known as rituximab from Roche, was approved in EU, not for MS, but for treatment of non-Hodgkin lymphoma, cancer treatment. So this is an anti-CD20 treatment that deplete B cells. And this drug has become very common now in MS as well, as we will see. In the year 2000, the Swedish MS registry was officially launched by Professor Jan Hillert. It has today a coverage of 24,000 patients, and that is actually more than 85% of the total Swedish MS population. And the success of this registry is partly due to that it is based on a decision tool to enter and also visualize data on individual patients for the doctors. So they get a visual feedback and a motivation to use this tool and to enter data into the registry. In 2004, the first what we could consider to be a high efficacy treatment was released by Bayesian. So it had a much stronger effect than the previous drugs, but it was considered to be a second line of use due to a risk of a rare brain infection called PML. Then in 2006, Anders Svenningsson, he lived in Umeå, a small, not so small, but a city in the northern part of Sweden. Back then, he treated a patient with Mabtera. So that was the first MS patient in Sweden that was treated with Mabtera or rituximab. So he treated this as an off-label drug and saw that it had a really good effect. So it was considered to be a high efficacy treatment. And this was really the starting point for the success of using rituximab as a treatment in Sweden. In 2014, another treatment, Tecfidera from Bayesian, was approved. This had a moderate effect, so it was better than the interferons from the 1990s, but it wasn't considered a high efficacy treatment. Since the rituximab was approved for this cancer treatment, but not for MS, and Roche wasn't really interested in doing a clinical trial either because they were going to lose their patent. Anders Svenningsson went on and got funded by the Swedish Research Council to do a multi-center phase 3 randomized controlled clinical trial of rituximab in Sweden. So he compared rituximab with 98 patients versus 97 patients on dimethyl fumarate with a 24 months follow-up, with the primary endpoint being the proportion of patients with at least one relapse during the follow-up. So they found 3% of the rituximab patients to have a relapse and 60% of the dimethyl fumarate patients. So this was published in 2022 in Lancet Neurology. And they used, as Peter mentioned, the Swedish MS registry as an electronic medical record. In 2018, the use of rituximab actually reached 53.5% in Sweden of all new MS treatments. And it was also this year that biosimilars of Mabtera, rituximab, became available in the EU. The same year, Roche, through its daughter company, I guess it is Genentech released ocrelizumab or Ocrevus, which is very similar to rituximab, but it is a humanized antibody, so it has slightly lower risk of infections compared to rituximab, but both are anti-CD20 treatments. In Sweden, we use rituximab mostly. We have very few patients on ocrelizumab, but in other European countries, the ones that are part of the federated learning, in More-EUROPA, Italy, Denmark, and Czech Republic, they use more ocrelizumab. It's interesting to notice the price difference between the two. Ocrelizumab treatment costs around 18,500 euros annually, whereas a rituximab treatment may be something around 1,600 euros. So now we get to the More-EUROPA MS case studies. So study 1 and 2 is done by my colleague Elena. That is done only in using the Swedish MS registry. So it's a target trial emulation of refund using data from the Swedish MS registry, but after removing the refund patients. Then study 3 and 4 are looking into doing kind of the same thing, but with the other MS registries in Europe. So Bo in study 3 is looking at a traditional meta-analysis, and I am developing federated learning methods in study 4 for comparing rituximab, ocrelizumab, and dimethyl fumarate in these 4 registries as if data was pooled. So in study 4, I'm replicating parts of refund as a case example. So just again, and a kind of a graph showing how the refund study was set up, because this is similar to what we are doing then. We have a 24 months follow-up from study start of each patient, and we are going to count the number of relapses for each patient between these 2 study arms. But of course, since across these 4 registries, we don't have rituximab in all countries to that amount. So, and Sweden do not have ocrelizumab. So what we needed to do was to combine rituximab and ocrelizumab into one study arm that we call the anti-CD20 study arm. And then we have dimethyl fumarate as study arm number 2. The inclusion criteria then for this study is a little bit broader than the refund was. We allow an age range from 80 to 60, an EDSS less than or equal to 5.5. And the start of treatment must be between 2018 to 2021. So it is run in 2 parts. First, we need to run propensity score. We could either use matching, which would be that we pick out those patients that are similar between the 2 study arms and only use those. Or we could use weighting, where we put a higher weight on those patients that are similar between the 2 study arms. So we are doing weighting. So, and for that, we are using a logistic regression model that includes variables like age, sex, time to index from study start to give us a kind of a calendar year effect, whether or not they had a previous relapse, and then also country-specific variables and interactions with whether or not they had a previous relapse. So that is our logistic regression model to predict whether a patient belongs to study arm 1 or 2. Once we have run this logistic regression model and obtain the weights, we can run the next step, which is the same thing as I did in refund, is to run a log binomial test on the differences in relapse activity between the 2 study arms during a 24 months follow-up period. But what we have done so far is to run this logistic regression model using federated learning, obtaining the weights, and then look at what is called standardized mean difference between those variables before and after applying the weights to see if it works or not. So in federated learning, then a key question here is, how can we really run the analysis if we cannot merge data? So traditionally, we could, of course, do a meta-analysis. That is what Bo is going to do. Mariana will talk after my presentation about a very interesting approach, which is to use something called a Bayesian inference. There's also something now being developed called artificial intelligence models to create synthetic patients. Because if you can create a model of the patients to create synthetic patients, then you can share that synthetic data set instead between the countries. And then those are, of course, not real patients, so you can always share those and do the statistics. But my interest is in doing federated learning through something called distributed computing, because that is mathematically identical, actually, to running it merged. So how does that work? Well, to understand that, we need to understand how data are stored when pooled. If we can understand that, then we can separate the problem and see how it could work also distributed between computers, where each computer has its own data set. So I want to go back in time to 1961, because it was around this time that memories looked like this. So you can actually see each single bit of information on this memory. So this was called a core memory. And if you zoom in closely to this core memory, you can see that each little bit is built on 4 small magnetic cores. So these kind of memories, they were actually hand-woven by skilled women, crafted manually. So I'm going to show how that. So I have one actually here. So you can see in detail how it looked like. So this memory is then from 1961, more than 60 years old. It could actually, one such memory could store 128 bytes of information. And of course, in such a computer, you would have many of these stacked together. So if we then assume that we would have these old computers to store a patient registry, we could assume that, well, for this particular memory, we could store maybe 16 patients per registry. We would store Sweden first and then Denmark. Each row here on this memory would then be able to store 1 patient, and we could store information about age, sex, country, whether or not they had a previous relapse, and treatment. The information we need to run this clinic emulation trial. So now we understand how data is actually stored in a computer memory, and modern computer memories are similar in the sense that they are stored like sequentially like that. So now the next question is, how does it compute? How does it look at? Does the computer need to look at all this memory all at the same time to compute and run logistic regression? And for that, I want to go to 1966. This computer had actually the hardware, the actual computational units visible like this. So if you look here, you can actually see the transistors on the circuit board. So looking again at this, it looks like this. So this is a circuit board from that computer. And so you would have many hundreds of these in the computer, and then wires to connect them all. So what would such a small circuit do then? Well, this one could calculate 5 Boolean AND operations in parallel. So a Boolean operation, an AND operation in this case, take 2 binary numbers. If both are 1, it will output 1, otherwise it will output 0. And so if you have these so-called logic gates, then you can connect them all to calculate something more complicated. So this one would just be a small part of a larger circuit board, for instance, to be able to calculate the addition of 2 numbers. So actually in 1969, the development went even further, and you would be able to store the whole circuit board for adding two binary numbers into one integrated circuit, and it looked like this. I have it here as well. So this, as you can see there, says 7107A. That means this chip is from 1971. So this is more than 50 years old, but it's 10 years younger than this core memory that we looked at. So how does it work then? Well, it can only see two values at the same time. So say that we want to add 6 plus 11 to get the value 17. That would work like this in binary numbers. So we have 6 in green and 11 in red. We would put in the numbers in at the top of this circuit, and then it would calculate and give out the sum of the two. So here's an important principle in both computer science and in mathematics. You only need to calculate two numbers at the same time. You don't need to see more numbers at a given moment when you do the calculations. And that is a pretty important thing, because it means that you don't need to see all the patients all at the same time. So that makes it a little bit easier to understand how we can also then distribute computations. Now, this arithmetic logic unit that does these kind of mathematical additions, it's just part of a smaller part of the whole CPU. So how is it then used in a processor, a computer processor? Well, if we look at the Z80 processor, which looks like this, if you then the ALU is just a small part of it, and then it has instructions to add two numbers. Take two numbers from the memory of the computer, add them together, and then store them back again. And I'm going to show you this processor was very popular to use in old computers like this one. This was one of the first home computers in the 1980s, Sinclair Spectrum. But it was also actually used in calculators like this one. So the Z80 processor, I have one of those as well here. So here you saw where it was located in the actual integrated circuit. And modern processors, of course, they work in kind of the same way. They have billions of logic gates today, but the principle is the same, to calculate the sum of two numbers and then store them back into memory. Then in the 70s, we also got simpler ways of storing data. So here you have different kind of storage devices. You may find this a little bit more familiar. And of course, I have the results now from the More Europa on a floppy disk here that we will take a look at later. So moving on then, now we understand, hopefully to some level at least, how data are stored in a computer and how the computer can read data from the memory and use this kind of logic units and arithmetic logic circuits to do calculations. So how does logistic regression then work? So if you would sit in R, you would typically write something like this. You would write the GLM command and it will do it for you. But if we will have to do this ourselves, which we do if we want to develop our own federated learning, we can take this simple example. Say we have three variables here, age, sex, and relapse for each patient, and then whether or not they had treatment 0, which is drug A, or treatment 1, which is drug B. So in logistic regression here, for propensity score, we want a model that can predict whether the patient had treatment 0 or treatment 1. So we put in these four variables, age, sex, previous relapse, and also an intercept variable. We multiply each variable with its beta estimate, and we calculate the sum of those multiplications. That is then a linear regression. But the linear regression can then give values above 1 and also below 0. And we don't want that because we need the output to be between 0 and 1. And that is where we use this sigmoid activation function. So by taking the sum through this sigmoid function, we get values between 0 and 1. And what you see here is actually how logistic regression works. It is also the simplest form of a neural network, one without any hidden layers. And this is just another way of expressing mathematically how logistic regression works, with y hat being the estimated treatment. So y hat is the estimated treatment and y is the true treatment for a particular patient. So now the key question is here, across patients, what are the optimal beta estimates then? Well, we can use something called a cost function. It's a kind of the error calculation. So we calculate an error for each patient. If the beta estimates are really wrong, we will get a high error. It will not.
Lars, you're dropping out.
Hello, can you hear me now again?
Yes, you're back, Lars.
Thanks God. Okay, getting back then. I heard a blip in my headphone and then I realized that something strange happened. Did you hear me up to this point? So I didn't talk to myself for, I don't know, 20 minutes.
No. We heard you until you started explaining the error here.
Good. Thank you so much for that. Yeah. Okay. The error, yeah. So we can calculate an error for each patient given the beta estimates. And if the beta estimates are better, then of course we will get a lower error. So we can calculate that across all patients to get a cost function that is then for all patients. And we want to reduce this cost. And how can we do that? Well, we could, of course, start. We don't have any idea where to start, so we can just start with setting all the beta estimates to 0. If we do that and calculate the estimated output for one particular patient, we may find that the true output should have been 1, but the model says 0.2. So that gives a high cost value for this patient. And if we put that into a graph, we see that we end up quite high in this cost function graph. And what we want to do is we want to go downhill. So we can do that by calculating the derivative of the cost function across all patients. Then we know, kind of standing there in the slope with skis on, which direction to go. So we can calculate the derivative of patient 1 really simply by taking the estimated value minus the true value and multiply it with the independent variables. Then we get the gradient for the cost function for this patient. And then we can do the same thing by just taking the sum of them all across all patients. And then we can update the beta estimates like this. We take the beta estimate and we subtract the derivative of the cost function multiplied by a small scaling factor. We can then, once we have updated the beta estimate, we can iterate and do the same thing again and again until we reach a final beta estimate that doesn't really update any longer. So now we see that we actually get a good estimate for this patient. The true estimate was 1. The true outcome was 1. The estimate was 0.9. So how can we understand this now from the point of view of this core memory? So if we would store all the patients like this and have all the patients stored as rows, as I said, if the data was merged like this, we could just run the sum across all patients through these rows and then update the beta estimates. But since the change in beta is just a sum across patients, we can just split it up then. So we have Sweden separately from Denmark. So we calculate Sweden first and Denmark after Sweden, and we just add them together like this. We take the Swedish part and the Danish part, we add them together to get the final derivative of the cost function. This is mathematically the same as if we would just run it as one big sum. And if we can do this, of course, then we can just split it up into 2 different registries and let them calculate their own gradient. And then we need a central computer that can communicate with them and send then the beta estimates. They would calculate their gradients, send them back to the central computer. This is just aggregated level data. So no individual patients are being sent to the central computer. It would then update and iterate again with new beta estimates, and you would go like this for a few iterations. And we can, of course, increase the number of registries to 4 registries, and that is what we have in More Europa. So this is basically how logistic regression works when you distribute the calculations across different computers, each computer with its own data. Yeah, I should just mention briefly that the method I just described is called gradient descent for optimizing the beta estimates. There's a better method called Newton-Raphson that we actually also use because it is much faster. There is a package already available that is called DataShield, and DataShield is it's quite complicated to install. It can do things like logistic regression federated, but you have to install a Linux server at each registry with a quite complicated setup, as you can see on this image. So what we wanted to do in More Europa was to make it much simpler. So we made something called FedLite. This is just a working title because FedLite appears to be already taken, so I must come up with a better name. But the idea is that it must be simple to run. So each registry should only have to receive one R script that they run in R at their own computer, and then internet connection so they can send the gradients to a central computer. The communication should be done through existing file distribution services. We need a common data model. I haven't talked about common data model, but we have one in the BMSD that we are using. So we have a common data model, and we are using that one also in More Europa. And the case example is, as I said, logistic regression. Here's just another kind of overview of the process that we are doing with the model and the registries and how they communicate with each other. So the way we communicate data is through RPins. RPins is an R package that allows you to share data through either Dropbox or S3, Google Drive or OneDrive, and we use S3 at KI to share these aggregated level results. This is just an image of how the infrastructure looks like, with the conductor being responsible for updating the beta estimates and the different registries communicating with this conductor through this RPins repository. So what we have done so far is to run part 1, which is to run logistic regression to obtain propensity score weights for the two study arms. And so the next step is to run part 2, is to run log binomial and see if there's a true difference between anti-CD20 and dimethyl fumarate when it comes to relapse outcome during the 2-year follow-up. So when we ran part 1, we got a result after 6 iterations. When you do this kind of calculations, all the computers, all the registries need to be connected all at the same time, because the iterations is synchronized that way. But it only required 6 iterations, so the running time was 3 minutes. And now it's time to look at the results. So I have the results right here. So we will take a look at them. So I will put it into the disk drive here. And then I will load it in. So we will see if we find it on the computer here. So here we see a file called results. I will load that one and we will take a look at it. Takes a few seconds to run, but load, I mean, but it will be loaded very quickly. Okay, I need to load it from the other drive. Demo effect. Oh. Oh, I tried to load it again. Sorry, this part, I really want it to work. It worked. I tested it so many times and it worked before. Okay, let's see. I think I have it still. Okay, so here's the results then from applying the weights. So what I'm going to show you first, and I'm going to zoom in so you see it better. So on the columns here, we have the Swedish registry, the Danish, REMAS is the Czech Republic, RIS is the Italian. So here we see the number of patients that we had for the anti-CD20 arm, a total number of 8,135. And for the dimethyl fumarate study arm, we had 8,272. The mean age for the anti-CD20 study arm was 42.3. Whereas it was 39 for the dimethyl fumarate. So here we see an example of that. There's a little bit of a discrepancy here for age between the two study arms, one that we wouldn't get probably if we would do a clinical trial with randomized patients. So we can then calculate the standardized mean difference. If that is higher than 10%, we have a problem. We are very close at 9.6%. So hopefully when we apply the weights, we can get this number to become to decrease. Then we have the proportion of women on the anti-CD20 study arm, which is 64.9% in total and 70.2% for the dimethyl fumarate. And this is also a quite big discrepancy. I mean, if we calculate the standardized mean difference, then we see that this is now higher than 10%. So this is a problem. We want this to be reduced after applying the weighting. The time variable is time from study start to when the patient got the treatment. And we want this to be about the same between the two study arms, and that is very close. And we see that the standardized mean difference is also very low, 1.9%. Then finally, we have the anti-CD20, the number of patients that had relapses 2 years prior to studies to their own index date, which was 64.9% in the anti-CD20 and 70.7% for the dimethyl fumarate, and that is a standardized mean difference of 5%. If we now apply the weights, we can see how these are updated. So here you see now the total number of patients with as in effective sample size. So that is around 7,000 now for the anti-CD20 and 6,282 for the dimethyl fumarate. So it is a little bit lower, but it's okay. That is expected, of course. The anti-CD20 mean age, then, if we can get that to be more close to each other, that would be really good because we have a standardized mean difference of 9.6. So we get 40.9 for the anti-CD20 and 40.9 also for the dimethyl fumarate. That is really good. So we see that now the standardized mean difference is just 1%. This is a variable that we hope for the most because here the standardized mean difference is 14.8%. So we have 64.9 for anti-CD20, and we get it down to 67.9. And for dimethyl fumarate patients, that is also very close, 67.6. So the standardized mean difference now is 0.9%. That is very good. So we can continue like this, and for the time variable, we already had good results, but it is even lower now at 0.5%. Then we have relapse, and if we then update that variable as well, we see that standardized mean difference is now not 5%, but 0.4%. So that was the whole result that I wanted to show you. So, and that was the end of my presentation as well. So thank you very much for listening.
Thank you, Lars, and that was fantastic. Really enjoyed that thoroughly. I love to see. We'll make a screen picture of the results on your floppy disk. That obviously was good.
Yes, I tried to do the. I was afraid of getting a reader, and I actually got it, but then it loaded fine. So all computers, all software, all floppy drives. Yeah.
So one question, there are a couple of questions which I think we can address also afterwards, but the one that you really is burning is the one by Robert Borchas, my colleague, that really asks you why you are using a Commodore computer nowadays to calculate your outcomes.
This one that you see here is actually, this was not one of the questions I was expecting. But this one that you see here, let's see if I can show that to you. Anyways, disk drive. Okay, so this is my complete setup here. So I have the output from my Commodore computer right here. This is actually a replica of a computer that never existed. So this was supposed to be the Commodore 65. I do have a Commodore 64 as well, but it's much more difficult to set up this one in this kind of environment. But this is actually not an old computer. This is manufactured last year, and I'm not actually using it to calculate anything. It was just a fun thing to show you this way, how, you know, connecting the computer history with the history of MS and also an understanding of how computers work.
I thought as much, and I thought that's good to take that out of the discussion, that we're doing something very simple with something very old-fashioned. So no, definitely not. I propose that we keep the questions for now, the other ones, and that we will come back to them at the end of the session. So I would like to move to Mariana Jonker from Radboud University to take us into the Bayesian federated inferences. So Mariana, the floor is yours. You are still muted, Mariana.
Yeah.
Now I can hear you, yes.
I think you can see my screen now.
Yes, it is there, and it's in presentation mode. Perfect.
Okay, thank you. Yeah, so my presentation is about Bayesian federated inference. And this research is in collaboration with several people from the Radboud. Hassan Passira, he has left, unfortunately. Emmanuel Massa, Zoe van der Heuvel, and Tom Kolen. I already like to thank Zoe for making the figures in the presentation. So suppose there are multiple data sets at different locations available for analysis, and we like to do a statistical analysis using all the data. Then there are multiple possibilities, and probably the most easiest one is data sharing. Suppose there are 3 medical centers, each has with their own data. Then if they want to share the data, they send the data to one central location where the data are combined and this statistical analysis is performed. This sounds very easy, but in reality, it is often a problem. Medical centers are not allowed to share the data, and mainly the main reason is the privacy of the patients. So in practice, and this is even a bigger problem if the medical centers are located in different countries. So an alternative is federated learning, and Lars already explained a little bit about this, or actually more than a little bit. Oh, you don't see my slides?
They are not moving, indeed.
Okay. And now, do you see my second slide?
Now we're on data sharing, yes.
Yeah. Okay, then I will do it in this way. So an alternative way is federated learning. Lars already explained some details about that. Federated learning strategies have been around for some time, but they were originally not developed for medical applications. But nowadays they are more popular and often used in healthcare research. So what is it about? There exist multiple strategies, but grosso modo it is more or less like this. Suppose there are 3 medical centers, and each medical center has its own data set. Then in this center, a statistical analysis is performed. The results of that analysis is sent to a central server. At the central server, the results are combined and sent back to the medical centers. Next, within those medical centers, the estimates are updated, and the new estimates are sent back to the central server. So this is an iterative algorithm, and that continues until a criterion is met. If an extra center likes to join the whole project at a later moment, the whole estimation procedure needs to be redone. And especially this iterative characteristic of the strategy is sometimes a bit of a problem because of it's an iterative algorithm. It has to be done in a sort of a semi-automatic way. That means that you have to communicate with the medical centers multiple times, and results need to be sent across the firewalls of the medical hospitals. And because of that, we have been thinking about a new method that does not have this iterative characteristics. So what is this? So again, we have these 3 medical centers, and each medical center has its own data. Then within the medical centers, they do a statistical analysis. This analysis is different from the analysis that they do in the federated learning structure. And the results of that analysis is sent to a central server. And again, the results are different from the results in the federated learning strategy. At the central server, the results are combined into one global statistical model or result. So there's no, this is not an iterative algorithm. If a new center likes to join the project at a later moment, the analysis can be performed within the walls of this medical center as well, and the medical center can send the results to the central server. In the central server, the estimates are updated. So it's easy to include multiple centers at a later moment. Or it can also be used if the centers do not have the data available at the same time. So which steps need to be taken? So first, the medical doctors that do the research have to decide upon the medical research questions and which statistical analysis need to be performed to answer the medical research questions. After that, in every medical center, the same statistical analysis is performed. The results are sent to the central server, and in the central server, the results are aggregated to estimate a global statistical model. And this aggregation goes via a weighted average of the local estimates. And these weights are dependent on the local sample sizes and the accuracy of the estimates in the local centers. So a center with a large data set gives more weights to the estimates than a center with a small data set. With this procedure, we do not get exactly the same results as the results that would have been found if the data were combined before doing the analysis. But we get an approximation, and this approximation is often quite accurate depending on the sample sizes of the local data sets. So let's see an example. This is an example with the MS registry data that Lars already explained. So there is one MS registry in Sweden, but for this occasion, we split it up into parts. A part with Stockholm, with the patients from Stockholm, and a part with the patients from the rest of Sweden. So we have two data sets, or two centers. And we want to fit a logistic regression model with the outcome whether a patient has secondary progressive MS or relapsing remitting MS. And the variables in that logistic regression model should be age, sex, and the location. So whether the patient is in the Stockholm dataset or in the other dataset. And we want to do the estimation in a federated way. And these are the results. So here on the left, you see the names intercept, age, sex, Stockholm. These are the variables in the logistic regression. And next, you see the estimates of the regression parameters. So the first column is for the combined data analysis, where the data are shared. So all the data are combined into one database, and next is the statistical analysis is performed. So that is more or less the gold standard. That is what we want to have. The second column is the federated procedure, and that was already explained by Lars. This is a federated learning strategy, and in a federated learning strategy, the aim is to get exactly the results, the same results as a combined analysis. And that is also what you see. The estimates are exactly the same. In the third column, you see our estimates, the estimates with the Bayesian federated inference method. And what you see is that the results are almost the same as the results based on the combined data analysis, but there are some minor differences. And in the last part, you can see the estimates of the standard errors. And here, more or less the same story holds for the federated the estimates are exactly the same as for the combined analysis, and the BFI is a good approximation. It's a good approximation. So based on this, you can already conclude that at least in this example, the fed-light and the Bayesian federated inference method work perfectly. After developing the basics of the Bayesian federated inference, we were able to extend the methodology. And now, 3 years after we started the whole project, we are able to include situations about, for instance, if there are heterogeneous populations, so the populations in the different locations are heterogeneous, then we know how to deal with that. Or if we have heterogeneous models. But also if we have unobserved features. For instance, if in one of the data sets in the example, the age was missing completely in one of the centers. So for instance, in Stockholm, the age was never recorded. But we do want to include age in the data set, in the statistical model. And we know how to do that. We can also deal with many features, many covariates. If you have many predictors, for instance, now in the example, we have age, gender, and location. But for instance, if there are many covariates, then we always have to be careful with statistical analysis. Because if we do not account for this in a proper way, then we have to have the problem of overfit. And at the moment, we are working on a new project, and that is on federated causal inference, and a second one on small centers. So what if some of the centers have only 10 patients? Then analysis becomes more difficult because you have to do the local, you first need to do a local analysis. And now we already know, we have an idea how to solve that problem. But I'd like to explain a little bit more on the Bayesian causal inference. And that is also the next project that we are doing with Lars and his colleagues. So suppose we want to study the effectiveness of rituximab in MS patients, and we have observational data. If we have a clinical trial where the MS patients are randomized to a treatment group, a treatment rituximab, and a control group, a placebo, then statistical analysis is usually quite simple. After the patients have been treated for a while, you compare the treatment group with the control group, and if the treatment group is doing much better than the control group, you can conclude that the treatment helps. With observational data, this cannot be done. So suppose we have now I'm looking at a figure at the right. So this is a simple example of a cohort. We have only 4 patients in each group, in the control group, and 4 in the treatment group. In the control group, there are 3 gray people and 1 pink person. And in the treatment group, it's the other way around. There are 3 pink persons and 1 gray person. So if you give the treated, after a while, you can compare the 2 groups, the treatment group and the control group, and you may see that the treatment group is doing much better than the control group. From that, you cannot conclude that the treatment really helps, because maybe it is not the treatment, but it is the difference in the populations. So maybe the pink persons were much stronger than the gray persons, and that is the reason that the treatment group works is better than the control. So what you want to do is you want to do some weighing. And you weigh the persons in your data set so that the new pseudo population is balanced. The control group is similar to the treatment group. So in the control group, we need to have more pink people. So the pink person is upgraded or uprated, and the gray persons, we need less gray people, so the gray people are downrated. And in the treatment group, that is the other way around. The pink people are downweighted and the gray people are uprated. And here in this example, it's a quite simple example where you can see from the people whether you need to uprate or downweight persons, yeah? Pink or gray. But in reality, there are many more characteristics of the patients, and it's more difficult to decide whether, how to compute these weights. So that's why we use a propensity score model in order to compute these weights. And after computing the weights of the individuals, we do a weighted analysis. So an analysis based on the pseudo population. So in the federated setting, it goes like this. So we have these 3 medical centers again, and in every medical center, we estimate a propensity score model. These propensity score models are sent to the central server, and at the central server, the models are combined to one propensity score model. Next, the propensity score models are sent back to the medical centers, and in the medical centers, a new analysis is performed, a weighted analysis to estimate the treatment effect of interest. And these treatment effects are sent back to the central server. So in this way, we need 2 communication rounds with the medical centers, one for the propensity score model and one for the final analysis, the weighted analysis. But it still can be easily done. Today you have heard about the federated learning strategy and the Bayesian federated inference strategy. And these 2 methods work quite well in practice, but they are different. So what I did here is I made an overview of, in order to compare the 2 strategies. But I'd like to highlight some of the characteristics of both methods. So first, the accuracy. The federated learning strategy gives estimates that are exactly the same as the estimates in a combined analysis. But it needs an iterative algorithm. And the federated inference method gives an approximation, it's a good approximation, and it needs only one communication with the medical centers. Because the federated learning strategy is an iterative algorithm, it needs to be done in a semi-automatic way. And because there are multiple communications between the centers, usually the ICT department must be involved in this, and this can be a bit of an hazard. For the federated inference method, the methodology is quite hands-on at the moment. That means that if you work together with 3 or more medical centers, then the researcher, the main researcher, writes R code and sends this R code to the different medical hospitals for doing the analysis. So within the hospital, the researchers, the medical researchers, or maybe the statistician, needs to run the code him or herself and send the output by email to the central server. So this is a completely different approach from this perspective, but because we only have to send an email with some numbers. It is completely private. It's anonymous, and yeah, we don't need to involve the ICT department or the legal department for this. So what is my conclusion? I think, in my opinion, both methods work quite well, and which one you should use depends on the on the aim, the setting, and the research question. If you work together with multiple medical hospitals and there's one research question that needs to be, or several research questions that need to be answered, the BFI would be my preference, would have my preference, because it's quite simple. You communicate with the medical doctors see which results are sent back to the central server. So I think that is a very good solution. But if you work with multiple medical centers, and in all these medical centers, there are multiple PhD students who all want to do the research and all want to do their statistical analysis, I think it would be useful to spend some time on developing sort of a platform so that federated learning can be easily performed. So which method you should use depends completely on the setting. If you are interested in this research, this Bayesian federated inference research, I like to refer you to one of our papers. And if you are not a statistician, then maybe the blue one is most appropriate. Thank you very much.
Thank you so much to Lars and Mariana for your very interesting, very informative, and also very charming presentations. So I personally enjoyed it a lot. So now it's my time to try to moderate the questions. And I think I will just start with the one for you, Marianne. Even we have two that have been waiting for an hour. But Marianne, there's a question from Yanis, and he's asking, what is meant by many researchers in the comparisons of federated learning and the Bayesian federated inference methodologies?
Many researchers.
Yeah.
So in which perspective? In the last slide? So if you collaborate,
yes, in the table, I think in the table where you have the comparison of the methods.
Look up the slides.
Oh.
Okay. Yeah. So that was what I explained at my last slide. If there are many medical centers that like to collaborate, and in every medical center there are multiple people, multiple researchers that want to do their research and want to run their own analysis, the BFI is more complicated, because for every research question, for every statistical analysis, the people in the medical centers need to run code and need to run statistical analysis and send that to the main researcher. So that makes it more complicated. If you have a platform and more an IT and a nice platform where everyone can do the analysis, federated learning analysis without notice, and people, the medical doctors in the different centers do not notice that the research is done, the statistical analysis, then that for them that is much easier. So if a single statistical analysis needs to be done, or only a few, I think that the BFI works perfectly. But if you want to do a range of statistical analysis, and every day or every couple of more regular, then I think that is more a hassle for the researchers in the medical hospitals.
So it seems your question is answered, but does that mean more is like more than one? So like you have a simple case, a simple question, you would use one, and as soon as you have more, you would switch to the other methodology.
No, I don't think it is that black and white. It also depends on the medical researchers in the hospitals. If they don't mind to do some analysis on their computer, then it's fine to, and you can do multiple, you can collaborate with multiple researchers that do their own work. So it depends. It maybe also depends on who is in the medical centers. If it is only professors that with a busy agenda that have access to the data, then it's more difficult to collaborate than if there are PhD students who are willing to run the analysis.
Okay, thanks, Marianne. So then we have another question, and that is for Lars, and it's Helene that is saying, I heard that only simple calculations, so estimations with logistic regression, but not more elaborated models are possible with the federated learning. Is this really the case, Lars? And can you explain why that is?
Yeah, so I guess I don't know what kind of model she is thinking of, but of course there could be more complex models where you need to somehow interact with data from different registries combined somehow. I wouldn't know what that, but I can tell at least this: if you would do something much more complicated, such as neural networks, then at least the method that we have developed now would be just too slow, because you need to iterate this by updating the weights in a neural network. Then you need to iterate so much quicker, because it will take so much longer time to reach the optimal weight. So it will not be practical possible to do this iterative process synchronized between registries. In those cases, it would probably be better for each registry to run locally first for some iterations, then merge some intermediate results, and then you continue. But it's difficult for me to say exactly when it's not possible. We haven't come that far yet into, but for things like what we typically use, it's logistic regression, Cox regression, log binomial, for instance. We haven't had even methods for those to run federated before in a simple way. So that is where we try to, bridge the gap.
Okay, thanks, Lars. So there's another question for you, Lars. So all the products were taken in your example as single treatments. There were no combination therapies with synergistic effects for MS.
Exactly. So it's typically not. A patient typically in MS does not get more than one treatment. Even so, when they are switching treatment, there's often a washout period that they need to have first. And this may actually change now with the new treatments for secondary progressive MS, because some of those new BTK inhibitors may be good at slowing down the chronic inflammation that is causing a progression independent of those relapses, but may not be good at the acute inflammation. So then you might want to combine, say, rituximab with a BTK inhibitor. So that will be interesting to see now how we will do, because these BTK inhibitors are, I guess, have just been approved by EMA and will be started to be used now for patients with SPMS.
I have another question that you can maybe both answer. So you now explain these methods, and I know in one of your slides, Marianne, you said that this BFI methodology, it's a good approximation. So it's not as accurate, but it's a good approximation. So it leaves some uncertainty. So when you use this methodology for maybe answering a clinical research question, that might be okay. But what if you're using this methodology to answer a question from a regulator or potentially a payer that is not very keen on accepting a lot of uncertainty? How do you see this methodology used in those decision contexts? Do you think it is accurate enough, and what would you advise? And Lars, you can of course answer this as well.
Good question. Yeah, what we can do is we at least we can mathematically prove that if the sample size is large enough, that the results will be very accurate. But if the sample sizes are lower, then we have to deal with this extra uncertainty. That is true. Yeah. But usually in the examples we have seen is that they are quite accurate. You also have to take in mind that and the results in a statistical analysis are never exact, in the sense that it's always an approximation of the truth. And now we give an extra small uncertainty to that. Yeah, I don't know whether the EMA or the FDA will accept this methodology, of course. No, I don't know.
Lars, do you have a point of view on this?
No, but a follow-up question to Marianne on this one. So the uncertainty is of course one thing, and as long as you can still show a significant difference, say, between the two study arms, it's still, is it? So the question is then, is it safe to assume that there are at least no bias in a significant result?
Yeah, if we can assume that the centers are homogeneous, in the sense that the models, if we can assume that the models, statistical models in the different locations are the same, and I think that Lars also needs to make that assumption. And if the populations are more or less similar, also homogeneous populations across the centers, then yeah, we can prove that there is no bias in the estimates. Yeah.
Okay, I think it's a very interesting question. But I would definitely like to ask you more around this, but let's move on to another question that is sort of similar in nature. And that is like, do you have any suggestions to address, or would you have any suggestions to address federated analysis in the case of rare diseases, where typically the patient numbers per medical centers can be very low? So what is your view on this? And again, it's to both of you.
I can start. In case of rare diseases, it's always difficult to find enough patients. So every center, even if there are only 10 patients, every center can help in finding the evidence for new treatments, or yeah, whatever you want to do, study. And if you are not allowed to combine the data, then federated analysis is the only option. And then the question is, what kind of methodology do you want to use? A federated learning strategy or a federated inference strategy? But yeah, I think that in rare diseases, every center should be included in the analysis.
So Marianne, I actually have a follow-up question on that to you then, because Bayesian methods, I understood is a bit better at handling low numbers. than the frequentist approach that I use, for instance, for logistic regression. Isn't that true? And how is that then for your BFI as well?
Yeah. So what we do is that is ongoing research. But what we now want to try is that in the estimation strategy, the bigger centers where the sample size is high will help the estimation in the smaller centers. So you use information from the big centers, and you use this information in the small centers to estimate the parameters, and that can be done in a Bayesian setting. So you borrow information actually.
Okay. And I want to also add one other thing to this, is that, for instance, in Denmark, we have a past study that we run with to detect adverse events between a few registries in Europe for one of the treatments in MS. And Denmark has this problem that they are not allowed even to report if they have fewer patients than 5 with a particular adverse event, but more than 0, they are not even allowed to report that number of patients. And then I was asking them, well, but what if when we run federated learning, we don't have to see that in a sense? And because we had the same problem when we were going to run this with them, they wanted to make sure that the number of patients in a particular combination of variables didn't go below 5. And so I made a script for them so that they could see that, well, that wasn't the case in our study. But for rare events such as adverse event of some kind, say PML, which is very rare, then they wouldn't be able to participate actually even in that. It doesn't really depend if it's, they cannot merge, but they cannot even participate in federated learning actually.
Yeah. I was at a symposium 2 weeks ago. It was a symposium on federated learning, and there they used the rule of 10. So if there are less than 10 patients, then it becomes problematic. But still, yeah. Then someone asked, why 10? What is the evidence for choosing 10 or in your case 5? It's important that it is still anonymous. The information that is sent around or sent to the central server should be anonymous. And I still don't know whether there are really good guidelines for these numbers. And yeah, I
yes,
think
it's strange.
So there are a number of questions related to this, and one is, if the number of patients per center is low, is the statistics per center then trustworthy? And would you say this requirement that has to be 10 that has been proposed as the lowest number of patients, is that what you're saying, Marianne?
no, I don't think that the issue is whether that what you send to the central server, whether it's still anonymous. If you can you trace back the patients from that number that you sent to the central server. So it's not whether the statistics can be trusted, but whether it's still anonymous.
So what is the number of patients then per center that you would need to have a trustworthy statistics? Or if it's low, how trustworthy is the statistic?
Yeah, we don't know yet. So that is ongoing research, and we try to solve that with a Bayesian approach. And in small centers, we borrow information from the large centers in order to incorporate that information and to do a statistical analysis. So that is still ongoing research. So I can't answer your question.
Okay, so there's another related questions. What are your recommendations to address heterogeneity between the centers?
Yeah, in my case, I have a good example of that in More-EUROPA, because it turned out, now I can't show you those numbers, but it turned out that the proportion of patients with relapses that number was very high in Czech Republic compared to Sweden, something almost like 80%, the number of patients that had a previous relapse. One reason for this could be that in Czech Republic, they even so require for a change of treatment that they see that there's a recorded relapse of the previous treatment, for instance, whereas in Sweden, we don't have such requirements and can switch to rituximab for other reasons. And well, of course, they can do that too, but there's a discrepancy still here between the numbers. And one other thing was we may report less relapses in Sweden than in Czech Republic. So that is a kind of a heterogeneity between centers in terms of how different the patients may be. If the way I dealt with it, at least in the model, was to make sure that for a global propensity score model, I used interactions between the country variable and the relapse variable. And I think that picked it up pretty well, actually.
Marianne, do you?
With our methodology, we can account for different kinds of heterogeneity. At least on paper, the software is not there yet. In the software we have different. We have already programmed certain some types of heterogeneity, but certainly not everything. But in the theory is already there. The problem with heterogeneity is often that you have to think beforehand what kind of heterogeneity might be there and how do you want to solve that problem. But that is not really a statistical problem. That is a problem that you need to think about beforehand and in collaboration with the medical doctors. But a lot can be dealt with our methods. But you also need to be think of, do you want to do a federated analysis if the data are very different? If there is a lot of heterogeneity, then why do you want to do a federated analysis? Then maybe it's better to do the analysis locally and compare the results. So what do you win by doing a federated analysis if the data are very, very different, or the populations are very different?
So what if the populations may be not so different, but the data availability might be different in terms of different, like you said, what was it, in one of your registries, age was not recorded? So would there be an advantage in fitting separate propensity score models to each of the sites rather than using one model? Do you think that could be an approach?
Yeah, certainly. If the populations are very different, then maybe it's better to estimate the propensity score models in every center separately and use that to calculate the weights instead of estimating one global model that is used for estimating the weights.
Good. Lars, do you want to comment on this or?
No. I'm very happy with Mariana's explanation.
Okay, so then there's a question here. A lot of this was related to efficacy or effectiveness. So you looked into effectiveness. Are there any key differences in the applicability of the approaches if you used it to focus on safety instead? Or doesn't it matter? It doesn't matter what parameters you look at, or do you see a difference?
So, so you mean if I would, instead of looking at effectiveness, look at the focus is on safety?
Yeah, that's a question here.
Well, safety is a very difficult thing and very complex for us to run, and we do run them nevertheless, because we in Sweden rely on the patient registry to look at as a proxy of adverse events. So that means we can link the MS registry with the patient registry, and then we get information of when different patients were hospitalized, for instance, or either as an inpatient or if they were an outpatient. And we get the ICD-10 codes. But then we have to look at these different ICD-10 codes and group them in terms of various adverse events. So we don't just have now one outcome, like a relapse is just one outcome in the refund study, a very simple study to do in that kind of statistical sense. But here you have multiple different kinds of adverse events, some more serious than others. So that is, of course, a quite difficult and different kind of analysis that needs to be done then.
Marianne, do you want to comment on this, or you're fine?
No, I think I agree. Yeah, maybe one thing. If you are more focused on safety, then you still need to do some statistical analysis, and which statistical analysis you want to perform depends on the outcome. How do you measure safety? So I think it can still be done. Yeah.
Okay, then there's one more question. So it's about the federated analysis. Can you compare the federated analysis with the results of each registry independently? And could this help detect confounders that are not initially apparent? Lars, Mariana.
Exactly, it goes to both. I can start this time then. Yeah, well, of course, it's a good idea actually in More-EUROPA to let each registry also run it locally, and I have actually, it would be very interesting to see how that result would then potentially differ from running it federated like this. And maybe there would be things that we suddenly see that we haven't thought of. It's very difficult to figure out beforehand what that would be, but it is a good idea. So I will actually take that with me from today's webinar and think about that as well, that we run it locally at each registry too.
And we are already doing that. So all the centers has to do the local analysis first and send the results to the central server. And in the central server, we already compare the different estimates and see whether they are comparable. And if they are very different, then maybe you should not combine them. But we never have thought about using these results for finding unmeasured confounders. So yeah, maybe that is an interesting thing to do.
One thing here would be, for instance, say you have two different registries, Sweden and Denmark, for the refund. And it turns out that for some reason, patients in Denmark would be, say, younger on average than in Sweden. They can still run the analysis because all their patients would be younger, for instance, whereas in Sweden they would all be a little bit older. Well, if we would run a global propensity score weighting and a global log binomial, then we would make sure that we only compare patients that have the same age range between the two countries. But if we run it locally, then we are not controlling for that. So we would get one result for the Danish population that gets rituximab versus dimethyl fumarate that are younger, and then for Sweden that are old, but the end results may then be a little bit different. Could be. This is of course very hypothetical example of age, because that isn't the reality. But you could think of some kind of other variable, of course.
Yeah, may I comment on this?
Sure.
But if the age distributions in the centers differ, then the regression parameters in the model can still be the same. So even if the populations are slightly different, it might be helpful to combine the models. Doesn't mean the regression parameters are different.
Exactly. So you can there's still an overlap in my hypothetical example between Denmark and Sweden. It says that when you then run locally only, then you will not look at the overlap of age. Then you would look in Denmark at a slightly lower age range and in Sweden at a slightly higher age range. Then you will get a heterogeneity. But when you combine them through federated learning, then you would take into advantage that you can inference on a common age range that they both have. That is what I was thinking about.
And even if the ranges, the age ranges are different, it may even improve the accuracy of the estimates, because you have finally have a larger range of ages. Improves accuracy.
That too, yes.
I have one little question, and then I will hand over to Peter to close our very, very interesting session. So now you are looking at different registries, but could you combine other data sources?
You mean like we already do this in
I mean,
both
like,
Denmark and Sweden with patient registries, but maybe that isn't what you're after.
Could you combine it with, let's say, trial data?
Well, that is so yeah, I guess you could. I mean, if you have then, if you mean you want to run, say, Sweden and Denmark, and then some kind of database with clinical trial data as a third source of data.
Yeah, let's say you have done a single arm trial in China.
Yeah.
Could you
Very interesting example, yes. Yeah, I guess. What do you say, Mariana? Have you any kind of experience with that? It's a kind of a different data source where it's not real world data any longer, but a really, much more.
Yeah, we have some experience with that. It is possible to do that, but you have to be careful with the statistical analysis, because the statistical analysis should be different in the different centers, depending on how the data were collected. And also, if you want to combine the results, you have to take into account that the estimates may estimate something differently. So in a trial, you estimate an effect between a causal effect already, but in the other centers where you have observational data, you cannot just compare the two groups. Then you estimate something differently, and combining makes it more difficult. But it is possible, but you have to take the differences into account.
Very interesting. I think we're running out of time. Peter, I want to turn it back to you to close our webinar for today, and thanks a lot.
Super. Thanks, Marlene, for taking that question and answer session, and actually also really bringing the questions together. I think, Mariana, Lars, really amazing what you did today. I think it's a very nice way of presenting us with a better understanding of what federated analyses are and how we can do that cleverly, and how that is a bit different from what we usually do. I think we even got some extra research questions for the project, which is fantastic. You put people a little bit on the wrong foot with your Commodore computer, but I think that was really intended. So fantastic, Lars, and even despite a small hiccup halfway, I think we went smoothly through that session. So thanks all for listening. Thanks for the questions in the chat. Please keep following us. Where we're coming back in about half a year time again with another webinar. But we're also very happy to engage with anyone outside of the More Europa project. So thanks for today. Thanks for the attention, and especially also thanks to Samantha Sullivan in the background, who did a lot of work in getting everything working. That's it for today. Hope you have a good day, and we'll see you perhaps in the future. Thank you.
Thank you.